Okay, so the properties of genetic material DNA versus RNA both act as genetic material, but DNA is the dominant one. So RNA world, before DNA, RNA was the first genetic material. So there is like Watson-Crick model for semi-conservative DNA replication. Okay, replication, semi-conservative DNA replication. Okay, why it is? Because after the completion of replication, each DNA molecule would have one parental and one newly synthesized strand. So this scheme was termed as semi-conservative DNA replication. Now the experimental proof by Mizelston and Stahl, E. coli growing medium. So Mizelston and Stahl experiment. Okay, now you see in the first diagram it is 15 and 15 DNA. Okay, gravitational force so heavy showing N15 N15. Now after 20 minutes you see there is in the medium of N14, that is light one, and N15 heavy DNA. So N15 DNA, N14 DNA generation 1 after 20 minutes. Okay, so this kind of like hybrid formation, you see. Now it's come closer to the end. Now separation of DNA by centrifugation. Now after again 20 minutes, so total 40 minutes. So generation 2 like N14 DNA, N14 2 bands, and then hybrid 1 2. So you see light and hybrid both are showing. So after first generation only hybrid, now after second generation it is light and hybrid. In first generation it was heavy, but in second generation, now in the first generation and the first one, the only difference that it becomes little hybrid. So this is what we understood from this diagram. Now another, the machinery and the enzymes. So DNA polymerase, DNA dependent DNA polymerase, this is the main enzyme. There is formation of replication fork that can be continuous, discontinuous, and DNA ligase, origin of replication. See the replicating fork, you see template DNA, parental 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 strands like 3' to 5' 5' to 3'. Now the DNA polymerase acts 5' to 3' synthesis, right? So 5' to 3' synthesis, you see in from the bottom, you see when it is 3' to 5', so 5' to 3' synthesis, it is continuous synthesis now. So on the other strand, you see on the left hand side, so 5' to 3', so in the 5' to 3', it's like so always 5' to 3', so it's kind of opposite. So it is discontinuous synthesis, and on this right hand side it is continuous synthesis. So newly synthesized strands joined by Okazaki fragments by DNA ligase. So this is the replicating fork. Okay, replicating fork. Now transcription. What is transcription? The process of copying genetic information from one strand of the DNA, not the both the strands, but the one strand of the DNA into RNA is termed as transcription. Now the transcription, you see, now it has a transcription unit. So a promoter, the structural gene, a terminator. Transcriptional unit, a promoter, structural gene, and a terminator. Now DNA dependent RNA polymerase, this is the enzyme. There are template DNA, template strand, a coding strand, two types of strand. So schematic structure of a transcription unit, you see it's like coding strand and template strand on the top. So you see the structural gene between promoter and terminator. So at the the coding strand at the 5' end, it is promoter. At the 3' end, it is terminator. This is the transcription start here. Transcription start site at the promoter. Okay, so this is a schematic structure of a transcription unit. So template strand and coding strand. Okay, so this is a template strand, coding strand. Can you okay, now this is it. Now the promoter, terminator, and the flank the structural gene. The promoter and terminator flank the structural gene in a transcription unit. That's it. Now the transcription unit and the gene, you see the transcription unit and the gene, there is a term that is cistron, a segment of DNA coding for polypeptide. Okay, now structural gene can be monocistronic, polycistronic. Okay, now there now coding sequences, these are exons. Non-coding sequences, introns. So exons and introns. Monocistronic, polycistronic, and then cistron, a segment of DNA codes for polypeptide. Now types of RNA and the process of transcription. First one, initiation, elongation, termination. So initiation involves sigma factor at the promoter side along with RNA polymerase. It is the initiation. This is the RNA helix. Now elongation, when RNA elongates during between the promoter and terminator along with the sigma factor. Now termination, when RNA leaves the sigma factor and joins with the rho factor, RNA polymerase plus rho factor acts as terminator. This is the process of transcription in bacteria, not in the eukaryote but in the bacteria. So there is termination of transcription. This results in termination of transcription. Now initiator factor, that is sigma, and terminator factor rho. RNA polymerase is only capable of catalyzing the process of elongation. It associates. So cell capable of only elongation, but with the associations for initiation factor like sigma and termination factor to initiate and terminate the transcription respectively. So this is now the process of transcription in eukaryote. So it was in the bacteria, so it was in the eukaryotes. Now you see in the first diagram, so 5 to 3 to 5. So first step is the capping. So this is the splicing. So when there is formation of mRNA, now there are additional steps. You see, at the 5' it is methylene guanosine triphosphate, that is the capping, and then polyadenylation, that is tailing, poly tail at the 3' end. At the 5' it is capping. This is the splicing. Now mRNA heteronuclear RNA, hnRNA. What about hnRNA? First one, in hnRNA contains exon and introns. Exon coding, intron non-coding sequences. Now RNA splicing, that there is the removal of the introns that are intervening sequences. Now formation of mRNA. So this is messenger RNA, that is mRNA. So 5' to 3'. The process of transcription in eukaryotes. So this is it. Now the RNA polymerase 1 transcribes rRNAs. rRNAs can be 20S, 80S, 5.8S, where RNA polymerase 3 is responsible for transcription of tRNA, 5S RNA, and snRNA, small nuclear RNAs. So tRNA, 5S rRNA, and snRNA, small nuclear RNAs. Now RNA polymerase 2 transcribes the precursor of mRNA, that is the heterogeneous nuclear RNA, that is hnRNA. So this is it. There is term splicing, where the introns are removed and exons are joined in a defined order. There is capping and tailing. So capping is unusual nucleotide methylase guanosine triphosphate added, and 5' end of is tailing adenylate residues 200 to 300. So RNA world, you see the presence of introns is reminiscent of antiquity, and the process of splicing represents the dominance of RNA world. This is it.